Welcome to Physbriology, where the teacher becomes the tutor. Today we're going to be talking about the very basics of glycolysis. Now this tends to be a process that is extremely overwhelming to students, especially with rate-limiting enzymes and all the sub-steps. It can be a little confusing and a lot to take in. So what I did in this video is I broke it down into its very basic components, because as we can understand the basics and gain confidence in that, it's easier to add in those details. So when we're talking about glycolysis, it is going to be how the body utilizes our carbohydrates to create energy. So we have two different types of carbohydrates in the body. We have the stored form, which is going to be glycogen, and the non-stored form, which is glucose. So if we are pulling from a stored form to be glycogen, then we're going to create glucose. So we'll always convert glycogen into glucose in order to start glycolysis. In some cases, we'll use the unstored form of carbohydrates and start just with glucose. So when we are breaking down our carbohydrates, again, if it's stored, it's going to start as glycogen, be converted into glucose. If it's unstored, it will just be glucose, and we'll start here. So glucose goes through a series of reactions. There's a rate-limiting enzyme in there, phosphofructokinase, which we don't have to worry about for this video. And glucose is simply broken down into two molecules of pyruvate. Okay, so when glucose is broken down into pyruvate, our net or total ATP that we are going to gain is 2. Okay, so again, glycolysis, we start with glucose. It's going to be broken down into 2 pyruvate molecules, and as a result, we are going to create a net of 2 ATP. Now with pyruvate, it kind of has two fates, and it depends on the presence of oxygen. So if oxygen is not present, so O2 is not present, these pyruvates will then be converted into lactate. The pyruvate will convert into lactate. This is going to be an anaerobic glycolysis. Okay, so ana is without, aerobic is oxygen. So anaerobic pyruvate will be converted into lactate. Now with aerobic glycolysis, what's going to happen to pyruvate is it will go into the mitochondria and be converted into acetyl-CoA. This is the beginning of a Krebs cycle, and I'll have a separate video for that just to keep these short and simple. So again, if oxygen is present, O2 is there, it's going to be aerobic in nature, and pyruvate will enter the mitochondria and become acetyl-CoA. Now, kind of a silly but helpful way to remember this is think of oxygen as your ticket into the mitochondria. So let's pretend like the mitochondria is the Super Bowl. If you don't have a ticket, you're not going to the Super Bowl. So similarly, if you don't have oxygen or your ticket, you cannot enter the Super Bowl or the mitochondria. So again, it's kind of a silly way, but it is effective in helping us remember. So one thing to note is in the process of converting pyruvate to acetyl-CoA, it's kind of a sub-process called the PDC, or pyruvate dehydrogenase complex. All right, super fun term. And in that process, we're going to create 5 ATP. So this is technically separate from glycolysis. Glycolysis, just know your net or total ATP produced is going to be 2. And when pyruvate is converted into acetyl-CoA, known as the PDC or pyruvate dehydrogenase complex, we're going to create 5 ATP. That might be important to note just depending on your textbook and how they do the total ATP count at the end when you go through the Krebs cycle and the ETC. So that is glycolysis simplified and in its basic form. If you have any questions, feel free to drop them in the comments, and we'll make sure that we get those taken care of.